So chapter 4 discusses genetics, development, and plasticity. I think genetics, evolution, development, and plasticity. And we're going to start with the genetics and behavior. So in many psychology classes, we get into this nature versus nurture debate, or really it is a discussion of how these things interact to shape human behavior. Historically, we have had people, proponents of this very genetic, we are born with who we are, our behavior, our personalities, and so forth. And other people, different people who were saying, no, it's about environment, it's about learning and training, from especially in those early days when the brain is developing, but it's all environmental. And of course now we know, and even as far back as in my day, we realize that this is an interaction, and the issue or the question now is how much each of these factors play in the role of shaping our behaviors or aspects of our personality, our cognition, our cognitive abilities, our perceptual abilities, and so forth. I have this question, as I told you, I'm going to keep asking my green questions, even though there's nobody here to answer me, which is a little bit lonely and not making me happy. It's the way it is. Examples of characteristics. You might pause and think about that so that you're actually using your brain to be curious and getting that brain active. And so almost any characteristic that we can discuss, from musical abilities, athletic abilities, personality characteristics such as neuroticism or openness to new experience, and all of what we can talk about, really. There are some characteristics that are not about genetics and environment, but for the most part, I've rarely had people say anything that didn't fall into yes, it is. We are looking at the interaction of genetics and environment, and asking how much influence does each factor have. When we're looking at facial expressions, we're going to look at this again quite a bit when we talk about emotions, but this is showing the facial expressions by people who are congenitally blind, so who are born blind, on the left sides, and then their sighted relatives on the right sides. And you can see that the kinds of facial expressions and how they are created exactly with our face appears to have a genetic contribution. At least this implies a pretty strong genetic contribution to facial expressions. So discussions of genetics often start out with Mendelian genetics. This 19th century monk Gregor Mendel told us a great deal about genetic inheritance with his experiments on pea plants. Told us about dominant and recessive genes, and really demonstrated that inheritance occurs through genes. Genes are aligned along chromosomes, these strands of genes or strands of DNA, and they come in pairs. So we'll talk about alleles of a particular gene. So if we look at the number 1 pair there in the picture on the bottom, which is showing our 22 autosomal chromosomes, and then our XY, in this case a male's sex chromosomes as well, that if we're looking at the number 1 there, there's going to be a place on the left one and a place on the right one that kind of match up there, and those will be alleles of the same gene. And I'll go ahead and talk from the picture on the far right, which is from a Psych 101 textbook, but I always really liked the way it showed everything and magnified everything clearly. If we're looking at a cell, and we're in the nucleus of the cell, in the nucleus we're going to have our chromosomes. We go and we magnify that and see the chromosomes, and then we take a little piece of chromosome, we magnify that, and we can see the strand, that area of the strand of DNA. So I'm going to walk through some of that again in a little bit more detail with words on the slides. So we have our genotype, which is the set of genetic instructions These are composed of those alleles, which can be homozygous, so coding for the same trait, versus heterozygous, coding for the trait differences in the trait, so they are different from each other. And then we have our phenotype, that is the observable characteristics of those genetic instructions. So we get gene expression converted into the features of the living cell. And so this will depend on whether or not we're heterozygous, and it'll also depend on our environment, potentially, depending on the gene. A gene is a portion of a chromosome, and it is composed of deoxyribonucleic acid, what we usually shorten to DNA. The DNA, that then serves as a model for synthesizing ribonucleic acid, RNA, and the RNA is a single-strand chemical that can now serve as the template or for the model for protein synthesis. Okay, and our body is made up of proteins. If I didn't say it in talking about the brain and how it works, eat your proteins. Try to get the amino acids that are going to be better for you in those proteins that you are eating, because we're utilizing them in this protein synthesis, in this taking of the RNA and synthesizing protein from it. The DNA becomes mRNA. It's actually messenger RNA that is then used for protein synthesis. And I show a picture here. It's kind of, if you can kind of see the right hand picture magnified again from the nucleus of the cell, this strand of DNA taking some piece of that is messenger RNA that is going then to be used for protein synthesis. And because I cannot show the kind of animations that other people show, I'm going to put the link in for that, for what looks like a link there at the bottom. That is a link at the bottom, and that'll be included with these slides. You don't need to know everything in that, but it explains, if all of this feels mysterious, this explains it with animation. It's very helpful. So proteins really determine the development of the body by forming part of the structure of the body and by serving as enzymes, biological catalysts that regulate these chemical reactions in the body. So I'm repeating some of what was just given in that video, but I'm just taking what I need for for where we're going to go with this. So this transcription looks like that DNA is partially unwinding. Okay, and it is acting as a template for the RNA. And we have this kind of matching up or coding for DNA made up of these nucleotide bases, adenine, cytosine, guanine, and thymine, that then code for adenine to adenine, cytosine to cytosine, guanine to guanine, and thymine to uracil, or what's the U on the RNA. And then you can see we have these codons, so these 3 nucleotide bases that are then going to code for a particular amino acid. And of course, we know proteins are made up of these strands of amino acids, so we're coding for these amino acids, which then get linked together into a completed protein. So we have our 4 nucleotide bases coding for our 4 ribonucleic acid bases. And again, so I've just put it in a different way here. I don't know if you've already noticed this about me or I've mentioned this, but if I didn't understand a topic when I was first needing to explain it to students, I did some research to figure it out, what I needed to know so that I understood it well enough to feel comfortable with my knowledge. Genetics is not at all my background, and so I did a lot of research on genetics, and I was still pretty confused about how this all works. But so here's the same kind of thing with a different visual that is potentially helpful or not, and just this example of the protein that's composed. So if what I've coded for from my DNA to my RNA are these three bases, cytosine, adenine, and guanine, those three bases are going to code for the amino acid glutamine, and we're going to put these amino acids together to form a particular kind of protein in the body. So we're going to get back to this idea of Mendelian genetics and back to those pairs of chromosomes. If a person has an identical pair of genes on the two chromosomes, that is being homozygous for that gene. If a person has an unmatched pair of genes on the two chromosomes, he or she is heterozygous for that gene. And again, as I said with the previous slide with the picture, so these are alleles, and our alleles can either be homozygous or heterozygous for a particular gene. Genes can be dominant, recessive, or intermediate, although a lot of the times when we're talking about this dominant recessive pairing that you've probably heard of in your biology classes many times, it's often more complicated than that. Some examples here are eye color, where it's really very clearly intermediate, and the ability to taste phenylthiocarbamide, or PTC, which has basically a dominant recessive kind of pairing, even though, again, it's slightly more complex than that, but I'm going to stick with the PTC tasting. So if you can taste phenylthiocarbamide, it is this bitter substance, but some people just don't taste it at all. And it's a dominant gene to be able to taste PTC. So if I have a dominant gene, it's going to show a strong effect, and we're going to mostly see the expression of that gene, whether a person is homozygous and receives two of this, in this case, being able to taste phenylthiocarbamide, or whether it's heterozygous, where I have one gene that's allowing me to taste PTC and one gene that does not allow me to taste, so that I don't taste PTC. I'm going to end up tasting PTC. That dominant gene is what's going to be expressed. And a recessive gene is only going to show its effect when it's in the homozygous condition. So when I get two recessive genes for not tasting PTC, that's when I'm a non-taster, or I don't taste that bitter substance. And here's how that plays out, even though, again, I feel like most people have done this in basic biology. I like to go back through it with the picture. So I have this 25% chance of really being a non-taster, and I have a 75% chance of being a taster, but I can get there in different ways. So I can receive the dominant taster gene from my father and the dominant taster gene from my mother. I'm a homozygous taster. I can receive the dominant taster gene from my father and the recessive non-taster gene from my mother, and now I'm a heterozygous taster because that was dominant. I can also get the heterozygous, the non-taster gene recessive from my father and the dominant taster gene from my mother and be a heterozygous taster. Or I can be the 25% chance of being the non-taster. I get the recessive gene from my father and the recessive gene from my mother, and I'm a homozygous non-taster. Examples like PTC and eye color can be misleading, and I've already said even those dominant recessive, it's a bit more complicated than that. But these genes that are a single gene combination, so sometimes we talk about other things as well. So PTC tasting eye color, which is really more intermediate, but is a single pair of genes. There's sickle cell anemia, hemophilia. There are a couple other disorders that I sometimes end up talking about, depending on the example from the textbook, to keep it as simple as possible. But this is not, even this is misleading in looking as it's this single gene combination, whereas some characteristics are polygenic. And so I remember when I first started teaching this at Midlands Technical College, there were people saying, and we had just figured out the Human Genome Project really figured things out very specifically about what was on our genes a few years before that, and people were very excited about it, and it seemed like we were going to be able to explain our genetics. But many characteristics, and especially these characteristics that are these kind of psychological likelihoods, are polygenic. It's not a single gene combination. There are multiple genes. And just as an example, something that might seem kind of simple, and it's not one of those traits like, neuroticism or creativity or something that's intelligence, something that's very complicated, is that we have 180 genes just to code for our height. So some genes are expressed only partly in some cells and not others. We have environment can increase or decrease the expression of a gene. All of these aspects add a lot of complication to genetics, not just the polygenic, but all of this, which cells are expressing what and how much our environment is influencing genetic expression. So I'm going to finish up here a little tiny bit early with a summary slide because I went on to the next topic and I realized that I was going to go over, and especially because I'm pretty good at reminding myself that I owe you guys 2 minutes from last Friday, so I'll keep trying to make this approximately 50 minutes so that you can plan your 50 minutes for the class. But sometimes it'll go a couple minutes over. Sometimes I'll make it a couple minutes under, and I'll make it up the next time. So just where we are, we've gone through most of what I want to say about Mendelian genetics and how we inherit our genetics, how we inherit our genes. But we've gone through some more specific things here about DNA being transcribed into RNA, which then gets used as the template for protein synthesis, which again is making up who we are, really, our proteins. And we've discussed the difference between homozygous and heterozygous genes versus the dominant versus recessive genes, and then really started to address the fact that it is more complex beyond this homozygous versus heterozygous single gene pairs, dominant and recessive kinds of genes, and we will continue on from there.